So hello everyone, good morning or good afternoon, depending on where you are in the world today. I hope everyone is refreshed from the summer holidays. You have logged in to today's webinar by the Drug Safety Executive Council, also known as DSEC. My name is Brian Murphy, Manager of Business Development for Cambridge Health Tech Associates. Our topic today is Translational Biomarkers in Early Safety Assessment. And based on the responses we've received from this topic, we have once again confirmed that this is quite a hot issue in drug development today. We are scheduled to go 60 minutes for today's session, but we anticipate that future dialogue will be necessary after today's session. In a moment, I will introduce today's presenters and panelists, but first, allow me to share just some brief background on DSEC in case you're new to the community. The Drug Safety Executive Council is a free online community for leaders and enthusiasts in the field of drug safety. Our mission is to drive discussion and action towards the goal of developing better and safer medicines for patients. We do this through webinars such as today's, as well as discussion boards, blogs, polls, and much more. The DSEC website, which you can see there on the screen, was relaunched just a few months ago, and many new features and ways to interact with your peers have been added. So be sure to spend some more time on the site if you have not recently. Further, since today's drug safety experts exist in many areas, including biopharma, academia, CROs, and beyond, membership within DSEC is now open to all. And as always, we love your feedback on how DSEC can better serve the needs of all of you. And so certainly communicate with us. You're able to easily contact our community manager. Her name is Janine Holly, and you can contact her or all of us directly via the DSEC website. DSEC is organized and managed by Cambridge Health Tech Associates, or CHA. CHA is a life sciences consultancy that uses its unique collaborative model to improve the speed, economics, and effectiveness of life science research and development. CHA leverages its communities, consulting, and technology evaluations to help clients and the industry as a whole achieve their goals. One of the main areas of interest of DSEC members particularly, as further demonstrated by today's topic, is how new technology can improve drug discovery and development. And so it is CHA's goal to help worthy technologies, such as biomarkers of drug toxicity, cross the chasm towards widespread adoption. And many of you are probably familiar with the technology adoption curve seen there on the right, as well as the cross the chasm concept. But feel free to check out the book by Jeffrey Moore for more details on that. And of course, as you may have guessed, CHA is a sister company to Cambridge Health Tech Institute, or CHI, which is a leading life sciences conferences company. Another activity for which CHA is well known is our management of the Predictive Safety Technology Evaluation Consortium. And this consortium has one primary focus, which is to collectively evaluate and qualify new preclinical safety tools in a manner that maximizes knowledge gained and also minimizes cost. There are several pharmaceutical companies members in the consortium, and CHA has assembled a library, a shared library of compounds used to test the utility of each technology. Of particular relevance to today's topic, we completed a consortium project last fall to evaluate a tool called MetaMaptox, which is a metabolomics platform for early safety enablement, which was developed by Metanomics Health in Germany. 12 major pharmaceutical companies participated in that project, and a phase 2 project is planned to launch later this fall. And so certainly if any of you have interest in learning more about that, do not hesitate to contact us. The additional press clipping shown on the screen is for a project that's been sponsored by Chantest out of Cleveland, in which nearly 10 pharmaceutical companies are participating to evaluate a new method to directly predict a fatal adverse drug effect known as torsade de pointes. And this project is culminating at the end of this month with a summit conference at the venue of the Safety Pharmacology Society meeting, which is being held in Phoenix, Arizona. So now on to today's event. And it is my pleasure to introduce today's presenters. Dr. Ernie Bush is the Scientific Director for the Drug Safety Executive Council. And prior to joining DSEC, Ernie spent over 25 years in biomedical research, ultimately serving as Head of Non-Clinical Safety at F. Hoffman LaRoche in Nutley, New Jersey. Ernie has facilitated dozens of market research and technology evaluation projects run by CHA, giving him key knowledge into how new life sciences technologies make their way to the pharma market. Don Van Dam is the general, I'm sorry, Ernie, did you want to just say hi real quick, just make sure everything's working?
Yes, good morning, hello, and good afternoon, everybody.
Great, thanks, Ernie. Don Van Dam is the General Manager for CHA, joining the firm nearly 1 year ago. Prior to CHA, Don served in senior level commercial roles with Eli Lilly and what is now GSK, as well as leadership roles in laboratory, hospital, and other healthcare settings. Since arriving at CHA, Don has personally interviewed nearly 500 life sciences experts to gain insights into the main challenges and needs of the industry. Don, want to say hi?
Welcome, everyone, and thanks for attending today.
Thank you, Don. And I will moderate today's event. And what we will accomplish today is to begin by sharing some key insights gained through recent research into the development, use, and adoption of translational biomarkers. And this will be presented by Don and will be followed by some comments from Dr. Bush based on a recent study. The balance of the time will be dedicated to Q&A, including taking questions and responses from all of you in the audience. So we definitely want to make sure we spend quite a bit of time hearing from all of you. To submit questions or comments during today's webinar, simply type into the chat box at the right of the screen, and we'll field as many questions and comments as we can during the time permitted. And lastly, before I turn it over to Don, we will share the link to a very brief survey later on in the program. And we ask you to take just a couple of minutes to complete it so we can hear your feedback on today's webinar. And so with that, I'll send it over to you, Don. And let me just pass the ball to you, and then you can take it from there.
Great, thanks, Brian.
Okay, you should be all set.
Okay. So can everyone see the screen that says Research Highlights?
Yep, absolutely.
Great. Okay. So we did, as Brian said, I conducted numerous interviews across the industry, and I just thought as a background and preface to a more specific discussion around biomarkers in the area of safety, I would give you some context for how people are looking at biomarkers across the board. So this is mostly just sort of thoughts and then quotes from the people that I interviewed. So in regards to what is the definition of a biomarker, well, if you say biomarkers, it's a huge field, especially here in this conference. And just as background, I interviewed everyone basically at a Cambridge Health Tech Institute conference, our sister company. And this person talked about that you can actually see the differences. Sometimes if you say biomarker, you're talking about genes. Sometimes if you say biomarker, you're talking about proteins. And so if you say a biomarker community, which is what we're trying to create, is a community of people interested in talking about biomarkers as well as drug safety, it can include a lot of things. Since I am involved in therapeutics, I am more on the protein side. That was what this one person said. Another individual, the second issue in their mind in this whole area of biomarkers is the definition of the biomarkers itself, because it's a generic term that hides many things. For instance, you have the biomarker of toxicity, and that's evident, but you have also biomarkers of efficacy, and actually you can have many other kinds of biomarkers. So one of the other key issues that people raised was that there's more and more outsourcing of biomarkers and other assays than ever before. And I just wanted to give you some actual background on what that person actually said. And they said that they're seeing a lot more outsourcing of biomarkers or any assay. And they had recently attended a technology symposium that was held by a major pharmaceutical company. And at that symposium, they were exposed to talk after talk that was about outsourcing and working with CROs and things like that. And they actually indicated that that's the direction many pharmaceuticals are going with, whether it's biomarkers or any other type of technology. And one of the keynote speakers at the event from this pharmaceutical company, although not specifically talking about CROs, what they said was that, quote unquote, we have great science in-house at pharmaceutical companies, but there's also great science going on outside of us. And so therefore, this person who I interviewed talked about companies are looking to reduce their fixed costs and have more flexible costs. And when you look at the CRO, you have those companies that embrace new technologies and want to develop more assays and be more proactive in going after that business. And then you have companies who are slower to react and make those changes. So it's an interesting marketplace. And again, whether it's a pharmaceutical company, a CRO, or a technology provider, people are taking different approaches to the marketplace. One of the things that was mentioned by another interviewee was the whole issue around capital equipment purchases being restricted in the industry, and kind of what to do about that and how to handle it. So again, just to give you a few quotes from that actual person, they said that budgets are tight across the board at pharmaceutical companies. The industry has to spend more time justifying the purchase of various instruments, and so that the technology providers need to be able to help a customer perform return on investment analyses. And they had been recently talking to their colleagues, some other instrument vendors, and they were saying that although money is still out there, the money may not be available for purchase. And some pharmaceutical companies, in fact, have set up depots of unused equipment, and so people can go into their depot and look around and see what they want and use it, and obviously that's good. Then you can just recommission the old equipment, and there's still an aftermarket business to be had. But it's more for those large companies that have hundreds of instruments out there, and that kind of thing, versus smaller companies which might not have the resources to set up that sort of depot. So again, another interesting trend that people are talking about and implementing.
Hey, Dawn, right before you go to the next slide, I just want to direct everyone's attention. There should be a poll on everyone's screen to the right there. During today's webinar, we do want to, based on some of the topics that arise, we want to just poll the audience and kind of see what people are thinking as well. So if everyone could direct their attention there for a moment, we'd love to hear your feedback.
Great.
Sorry about that, Dawn.
Thank you. Thank you for reminding me of that. So what we're wondering is, when you refer to a biomarker, how do you describe it? So please answer that question while I go on and tell you some more findings from the research. Another key finding in the research was that people are developing assays on different types of platforms. And here it was mentioned that the challenge that you always find is people doing things a certain way because they always do them that way, versus looking at potential new technology to help them do something maybe different or better. So this is, again, both a frustration and an opportunity in those people that I interviewed, in that how do we look for standards, but how do we also move the science forward? In regards to the standardization question, it was even delved into a bit more deeply, people looking at standardized scoring for biomarker assays. And the quote is here: "I think it starts with pharma, and that I don't think they would like to see multiple assays with different cutoffs out there or different levels of how do you score this. So I think it would start with them, even if it's not their drug, that they would like to see that if they have a certain companion diagnostic tied to their drug, that if another one comes on board, that it's not going to be confusing to the laboratory, and therefore they are looking for more standardized scoring so that the lab doesn't have to use the test differently." So again, another trend that people are talking about and dealing with and trying to figure out how to handle. Another issue that was brought up was the issue of fast tracking of diagnostics, and how can we make this happen. And that issue is really tied to the fact that the FDA is allowing for new compounds that address unmet clinical needs to have accelerated approvals, but then the diagnostic is behind. So we have to catch up somehow, and how do we do that? So this is another thing that people are wrestling with and trying to figure out how to address in the marketplace. Many people talked about the fact that we need to find ways to lower the attrition rate of biomarkers and assays. It's more challenging to get the assay and what the right cutoff is because there's been so many. They felt the attrition rate is just so high. In this case, their company has worked on many companion diagnostics, and they're saying, I'm sure there's many other companies out there, and they turned out to be not clinically relevant. So their thought process or their question was, later then do you go back and you wonder if you picked the right cutoff, and maybe if you had picked a different cutoff, maybe then your assay or companion diagnostic would have worked. So it's something that people are thinking about, and I think actually it probably needs more dialogue and investigation in the industry. Another area that was a trend in the research was genomics and biomarkers. This particular person talked about, and we're integrating now as much as we can that are on the genome populations, and all of these can be used in the drug development process to assess or to understand what is the molecular mechanism to stratify populations by saying that you have this target, that it must be, that must be it or not be it. And for a given patient, considering his limitation, this can or cannot be touched by your drug and create biomarkers of both, therefore, toxicity and efficacy. So it's not just two-dimensional, it's now three-dimensional, looking at the person, looking at the efficacy, looking at the toxicity, looking at the biomarker. It just has become more complicated rather than easier, but hopefully better science is resulting in the end. Creation versus validation was another trend mentioned in this research. I would say the answer is very common, unfortunately. That is, biomarkers can be easy to detect, to create, but are very complicated to validate. So that's the main issue. Not only the creation of the biomarker, but after all, its validation. We've had some talks lately stating that the problem of validating biomarkers is, at least for drug usage, it's that you need to validate them, to create and validate them during the drug development process. And it's still not in the mind of the pharmaceuticals. At least this person thought it wasn't. And I think there is some kind of a fight between the pharmaceuticals that create the drug and in new kinds of industries that create biomarkers. And they still have to negotiate between themselves. I think that's the main point for biomarkers. So this, of course, was coming from someone who is creating biomarkers and not someone in the pharmaceutical industry. So it'd be interesting to get people's thoughts on that later on as well. The government and biomarkers. Okay, I think the first concern or challenge or thing of interest is basically where the FDA and EMEA are going to land with respect to things like the need for biomarkers. I mean, as far as I know right now, it's still fairly voluntary. A lot of these things are part of the voluntary genomics data submission to the FDA, for example. And I think there's a follow-on point to that, too. At some point, I think I'm curious to see where the FDA decides to issue basically policy and say, you know, we're going to require biomarkers of efficacy. We're going to require biomarkers even in patient identification. I mean, they can do that, and then if they do, that clearly has a very, very sweeping effect on the rest of the industry. So again, we have another poll question for everyone in the audience. How often do you employ non-FDA certified or validated biomarkers to assess the safety of therapeutics in early development? And we're just asking you to select the best answer there out of 4 different potential answers. So that's it for me. We'll kind of come back and look at the results a little later and talk about that in the question period. But I'll pass it back to, I guess, Brian to coordinate next steps and Ernie.
Great. Thank you very much, Dawn. And Ernie, I'll go ahead and pass the ball over to you, and you can run through just, again, some of the comments that came through in a recent study that we performed. And whoops, oh, you already have it.
Okay, thank you, Brian. Hello again to everybody. Can you hear me, Brian?
Yes, I can. You're all set.
I am going to advance the slide. Did it move?
Yes, it did, yes.
Display? Thank you. So as Brian introduced, and Dawn referred to it as well, we've done a lot of shall we say, big picture interviews with people talking about what are your issues in preclinical development, what could DSEC do, or what does the industry need to focus on in order to be more effective or more efficient or both. But we also had a specific project where we were commissioned to look at a very narrow question about biomarkers, where we did in-depth interviews with fairly senior people in the safety community, all from pharmaceutical companies, some large, some small. So we tried to cover a very illustrative range of opinions. And specifically looking at this question of biomarkers during early safety evaluation, And I'm going to spend just a little time to review the, shall we say, the highlights from that research, and then we're going to jump right into a Q&A period, and therefore we, again, really encourage people to go ahead and ask those questions, because that's where I think we can drive some real value for the participants. So not only have we done this specific survey, I will refer to our set of interviews or project, but I will also refer to the annual technology survey we do, since we do have a history of opinions on this area, so we can compare sort of the recent comments to the history. So I have to say this is all in a background of how people view biomarkers for organ toxicity evaluation, and it's always been one of our, ever since we started asking the community, which is roughly dates back to, shall we say, 2007, over 5 years now, about this biomarkers and toxicities has been one of the focal points that people really want to see developed. So this is not a new thing, and of course I think we all can relate, or at least anybody who's been around for a while can relate. This was really emphasized back in 2004 with the FDA Critical Path Initiative, and in their outline document they mentioned biomarkers something to the tune of 28 times. So there's clearly been a focus on this for quite a while, and it was a bit surprising to me to see that that biomarkers, even during our recent interviews, were still a major area of interest. The community still really has a want, a need, and shall we say their mouth is still watering over the concept of having useful biomarkers of organ toxicity early on in the development process. But there is a slight flavor adjustment in that people now are really looking for biomarkers that or at least systems that help them understand how the biomarker affects the pathway or the system biology or the system toxicology. In other words, more about the mechanism. Biomarkers that are more mechanistically and more relate to pathway information seems to be gaining a lot more emphasis, or at least a lot more interest within the community. And again, this is borne out by not just these interviews, but in recent surveys as well. And I like this next point. All those interviewed agreed that in theory, knowing the levels and profiles of various safety biomarkers will improve understanding of the mechanism and the relevance of the animal studies relative to the human clinical safety prediction. So the feeling is if you have a, quote, good biomarker, and what good means and what biomarker means, Don has already pointed out, there is some sort of really broad or varied perspective on this, but nonetheless, at least the driver seems to be the feeling that a good biomarker will really help relate to animal studies relative to human clinical data. However, I got to say that enthusiasm is maybe now balanced against the concern of how do you validate, and I mean scientifically validate the biomarker. How many compounds do you need? What kind of organs do you need to look across? How much clinical data do you need to correlate against? The whole area of biomarker validation, Don also touched on this, but it really came out in our interviews, people's skepticism that an individual company could actually do enough of a validation to really be convincing that this is the way forward. And I'm going to touch on this as we move forward as well. This point about what constitutes a good scientific validation of a biomarker, I think, has been probably the major sticking point in the adoption of biomarkers ever since the FDA first really started to push this in the critical path documents.
Hey, Ernie, I just want to jump in really quickly. Don had mentioned earlier, some of the questions and confusion on the definition. You just brought it up a second ago, but also someone chatted us through the chat portion, regarding the definition of biomarkers. And the question was, why not just use the NIH/FDA definition of biomarkers? Are you familiar with that, and would you be able to comment on?
Yeah, I think that's great. I don't really think that there's a lack of a good definition. What I think is there's a lack of a common understanding of that, or a common usage of that definition.
Okay.
And you know, what I hope that we're not so much here feeling like not having a good definition is helping people move forward. I don't really think that's the critical step. It's just that, unfortunately, when we ask people biomarkers and they start answering, it's clear they're answering it from different perspectives. And what is so, you know, that's somewhat to be unexpected. Some people biomarkers means you know, changes in gene expression ratios. Some people it means, as we ask further, some people it means changes in circulating peptides or proteins, and some it's actually a metabonomics or metabolomics question. So I don't think it's actually not having a good definition available. There is a good definition available. I think it's more about how people perceive it and what they want. There isn't any commonality in their view. So did I answer your question or the question,
yeah.
Brian?
I think so. Based on what was sent through the chat, I think hopefully that covered it. But I'm sure if that person has further questions, they're welcome to resend it through the chat. So thank you, Ernie.
Okay, good. One of the things we did in this survey was, of course, spent about a third of the time asking people, what do you do routinely? And by routinely, we mean what do you do more than 50% of the time in terms of the leads that come through in your early discovery programs. And to no surprise, what we found was that larger companies tended, on average, to be much more aggressive about their early discovery screening paradigm and safety assessment in terms of the numbers and tests and how often and how early they run those tests. And the other thing that we confirmed was the utilization of software prediction models like DSEC were actually have become very commonplace. Even you know, people, there's sometimes still some questioning about how predictive or how effective those models are, but they have become very broadly utilized and across the industry in both large and small companies. And we see this trend in a number of different areas, and it was really confirmed in this particular program as well. So based on that background, one of the things I should have put on the slide was that there are some things that you could call biomarker-like, or that were built on biomarker-type technology that are used routinely. For instance, these high-content analysis cell health parameter screening, where you're looking at certain biomarkers, if you please, of cell health. It's done very often, particularly in large companies, looking at profiles of these cell health parameters. The smaller companies maybe less so, since it is kind of expensive technology. But we saw very broad, I have to say, probably 80% of the respondents now do some kind of cell health profiling screening in a high-content analysis format, looking at certain markers for cell health. And also, we were a bit surprised at the number of companies that do off-target screening, what has some people refer to as SERP screening, very frequently and fairly early on. That has become adopted, and I don't know if I'd call that a biomarker or not, but it's clear that type of screening has become, shall we say, the norm. Although there was a difference in that particular screening effort between large and small companies, where smaller companies tend to do those kind of SERP or off-target screening much later in the process, maybe just before entry into humans. But if you one of the things we looked at was gauging. We have the interview process consisted of a long list of possible screens or possible preclinical safety tests. And I don't really want to review that now, other than say it's still surprising to me across the industry and between large and small pharma how many of those tests are run at different times. And some companies do very little, less than 20% of the available pre-GLP tests do they run? And some companies run a lot, so there still seems to be a very broad distribution, and therefore the use of biomarkers follows that same range. Some people do a little and some people do a lot. And there doesn't seem to be any, I'm going to say, consensus across the industry right now about how valuable is and how important are these preclinical safety screening technologies and the use of biomarkers at that time. Last but not least, I think, so I'm going to finish up with, was there sort of one gee whiz conclusion that came out of this focus set of interviews? And yeah, there was. And that was, I have to say, that the use of biomarkers in an in vitro setting, a lot of companies are promoting and developing tests to look at in vivo outputs, shall we say in vivo metabolomics or in vivo testing of serum levels, et cetera. But what people kept telling us was in vivo was really too late. They wanted more focus on biomarkers that could be used in an in vitro setting to really help them make safety assessment decisions, or at least ranking or qualification parameters much earlier on. And so I think if there's one thing I'd like to leave with, both from particularly to the vendors, was the degree or the extent to which people said we really would like to see more biomarkers developed for very early in vitro safety assessment. So with that, Brian, I think that's sort of the overview, both from a high-level point of view or a general point of view from Don, and from this specific project point of view about the extent of where biomarkers are and what people would want. And I'd like to turn it back to you for a general Q&A discussion.
Great. Perfect. Thank you very much, Ernie, and thank you also, Don. And as you just mentioned and was mentioned earlier, we'd like to spend the balance of the time, which is nearly 30 minutes, covering a handful of questions that we've sort of prepared, but also we want to make sure we address the questions that are coming through in the chat. And again, feel free to send your questions and comments through the chat there on the right of the screen. You've noticed also there have been several poll questions that have been put up. We will compile the data, just the aggregate data from those polls, and we'll be able to send that out to you guys, as well as I anticipate that we'll put that either generate a discussion in the discussion board on the DSEC website, or even create a blog post that you can all go to, just to see what your peers are thinking about as well. And so I guess with that, and Ernie, as you know, many of these questions will be sort of directed to you for some of your commentary. But we also, as I said, would love any comments from the audience, and also Don may chime in as well. So I guess I'll just move on to the first question here, and it's sort of been covered, but maybe just to kind of recap what we've stated so far. In general, what is the current state of pharmaceutical and biotech companies employing biomarkers in early drug development? Maybe you could give a couple points, and then we'll also look to the audience and see what comments they have coming in.
Sure. So I advanced the slide for you, Brian. I still have the ball. You might want to pass it back to yourself. So I'm going to answer this question additionally with some other insights. So it's clear that biomarkers of a sort are in heavy use in early safety assessment. If you look at the cell health parameter indicators, they're used across the industry pretty much, I wouldn't say universally, but a very high percentage. But on the other hand, if you look at, shall we say, the next generation of biomarkers, those things that have been developed and / validated as a result of, for instance, the Critical Path Initiative or other global initiatives, and the adoption seems to be slower than one would expect. And I think particularly that maybe the KIM-1 kidney toxicity biomarker, there are people that say they use it, but they use it very selectively, well, sort of on a case-by-case if they need it. That isn't part of their, KIM-1 hasn't really been adopted as a routine GLP tox parameter, shall we say, for instance, in spite of its many advantages over traditional kidney biomarkers like urea or creatinine kinase, etc. And when you try to dig down and ask for the reason for that, you really kind of get two answers, although neither one of them are particularly satisfying or very clear-cut. The first is the traditional markers that we've used for toxicity, including histopathology analysis, they're set into our systems. There's a lot of historical data. And they can be achieved, particularly in these clinical monitoring or serum or plasma clinical chemistry devices can be done for pennies a sample or less. And where some of the new biomarkers are quite expensive to measure in that sense. And so one of the things that's lacking, one of the points I want to make is, while there is good scientific validation for some of these newer biomarkers, shall we say, the cost-effectiveness validation is yet to be there. People are unconvinced that it's worth the extra money to measure these things, since they are going to get the existing data and as well as the histopathology. Is it really worth the extra money for some of these new biomarkers? And Don touched on this too. This cost-effectiveness question has really not been, I think, appropriately dealt with in the biomarker community. Yes, we've got some good biomarkers, but are they really cost-effective? Is it really worth the extra money? And I think that kind of data has yet to been widely either published or accepted. And the other point is, I do think it's this sense, and Don also touched on it, about what is the cutoff, or what are the target levels. There doesn't seem to be a consensus. Some of these biomarkers are only available from one vendor, and the vendor maybe produces some data, but there hasn't been the kind of, and particularly in the area of toxicology, we talk all the time about it. They tend to move as an industry, not as an individual company, and so there needs to be more convincing done that biomarker, or the way to utilize or interpret that data, has not been universally adopted or set in a way that everybody buys into it. So I think the two answers to that question or three answers are that there has been a lot of adoption, but what's really sending the whole things back is 2 points, which is the cost-effectiveness question, and then the universal adoption question, or the universal setting of the limits or the determination of the value levels is still, I think, an area of weakness. Okay?
Thanks, Ernie. And actually, I wanted to just do a follow-up, and this is stemming from a couple questions that have come through the chat. One specific from Modi, who asked, so based on what you just said, I mean, what are the advantages in toxicological biomarkers over the classic histopathology? So that was one that came through in a related question from Zong, who's online. And the in vitro test, if the in vitro test is important, how do we comment on translating or correlating in vitro test to in vivo situations? So we're all this commentary about kind of in vitro biomarkers. What are the advantages of that over a classic histopath, and how do we translate it to an in vivo situation? If you could please comment on that.
Yeah, so I think referring to your first question, there is a desire, or just let me reemphasize. So the first question is, do we look for biomarkers that will emerge before the pathological changes in the animal, or signs that will tell us more in addition to the histopathology? So I think the answer is clearly and resoundingly yes. People want biomarkers that do not take in vivo studies or tests that will tell them if there's a safety issue, that don't take in vivo studies where you get the histopathology in order to produce them. In other words, and it really comes down to that cost effectiveness. If we have to start to do an in vivo toxicology study, that's expensive. It takes time, it takes drug, it takes resources to do in vivo studies. And if we had, we would really like to see tests that don't take that kind of commitment to the molecule to give us an indication about the safety liabilities. So I think the answer to that question is clearly we have good ways of determining toxicology, traditional ways, using histopathology, etc. But those tests, it's not that they aren't good, it's just that they're not cost effective. You can't really run a 4-week tox study on 10 compounds in a program. It's just not cost effective. So the alternative would be, can we have value-added safety tests that can be done in vivo? So I think the answer to that question needs to be, we need tests, or that people want tests, or what people are willing to pay for is tests to give them safety insights, but give them to them very cost effectively and very early on during the sort of the lead optimization or the methylbutyl phenyl substitution stage, where you can start to get answers to that. And then moving on to the second question it says, how do you comment on translating or correlating between in vitro tests and the in vivo situation? And I think that's part of this, the point that I was raising. People have a real issue with what is the validation strategy and how well can you do that, and can any one company really afford to do that? I think people kept saying to us, well, we see validation or biomarkers proposed all the time, and they maybe run 5, 10, even maybe 20 compounds, but we just don't, 20 is not enough. We want to see much broader, much more robust validations that these biomarkers are actually predictive of clinical outcome, and would like to see them run on 100 or 200 compounds set. And when you, again, if you're talking about an in vivo situation, running a test across 200 compounds in an in vivo testing is prohibitively expensive. Where for an in vitro test, I think you can do that kind of validation and can begin to get that kind of confidence that your in vitro test is actually predictive of at least an in vivo outcome and maybe even a clinical outcome. So you think I addressed those, Brian?
I think so. Thank you. And actually, just based off your last comment, another question came through from Thomas in the audience. And I do want to remind everyone that certainly Ernie is addressing a lot of these questions, but if any of you have opinions or responses as well, definitely don't hesitate to send those through, and I'll be happy to read some of those as well to keep the discussion flowing. But back to Thomas's question, Ernie, you were just talking about validation, and the question from Thomas is that they distinguish between validated assays and validated biomarkers. And he differentiates those as that validated assays are tools for drug development, whereas validated markers are to be used for filing with agencies such as the FDA. Would you care to comment on that distinction? Or?
It sounds reasonable. I have to say, yeah, I'm not sure that in my mind, while there probably is a legal or shall we say a regulatory distinction there, I'm not sure that I actually would feel that scientifically that distinction is that maybe that profound. But I understand, and I maybe used in maybe too generic a definition of biomarker for what I was talking about. But I think the logic is the same both ways. You have some molecule you're following that you believe is indicative of some biological response, and therefore predictive in some way. And whether it's an assay or a diagnostic test, I think the logic is the same both ways.
Great.
But
thanks, Ernie.
As Thomas pointed out, it actually makes a lot of sense to me. So.
Okay, great. Sure, and if anyone else has comments from the audience, don't hesitate to send those through. We will get to a lot of more questions and comments that are coming through. I will move on to sort of the second question that we have prepared here. And that is, I believe on the screen, it says, how close is the industry from regularly applying in vitro and or in vivo biomarkers to assess early stage safety and efficacy?
Yeah, to me, the answer is they're already doing that, although maybe everybody doesn't acknowledge it as such. These cell health profiling tests are really just an application of a cell health biomarker to the screening paradigm. And some of these cell health biomarkers are, of course, just cell survival. They're very simple, and some of them are actually quite intricate, looking at certain redox balances within, say, the mitochondria. So I actually think that we could argue very successfully that biomarkers have been integrated into in vitro, and of course there's a lot of traditional in vivo biomarkers that we've done for years, if not decades now, as I talked about creatinine kinase or any of the liver enzymes, and then a relatively new one which has, I think, crossed over from being more exploratory to routine is, of course, the cardiovascular troponin biomarkers, which are used a lot. So there are good examples of biomarkers being used for safety assessment. On the other hand, I think where people, when they look at this, they're asking, well, what about the new ones, the ones that we haven't used for decades, the ones that, say, the Critical Path Institute did a nice job of collecting the data and submitting it. And there I have to say there is still somewhat, although we get answers to this, I can't look people in the eye and say how come these new ones haven't been widely adopted. But I do think the number one reason why there's still some dragging of feet maybe in the adoption of the new biomarkers is this cost-effectiveness analysis. Yes, these biomarkers have been scientifically validated, but have they been cost-effectively, or has there been demonstration that they're actually providing enough of an advantage to justify the cost and the effort to run them? And I'm not sure that data, if it is widely available, it hasn't been as widely accepted, but I'm not even sure it's as widely available. So I think the short answer to recapitulate is that we are actively using biomarkers in vitro and in vivo across the industry right now, but the process of actually integrating new biomarkers still seems to be somewhat problematic and slower than I think most people would have anticipated.
Excellent.
Okay.
Thank you, Ernie. I did want to just read a comment that came through from Bill in the audience. We had a question that came in and some discussion around histopathology and sort of the advantages of biomarkers over that. And Bill wanted to just emphasize that histopathology is not a translational biomarker. And he went on to say that, obviously you don't necropsy humans, but it is the touchstone for tox. In vitro tests can be placed before GLP tests and points at which the compound development has too much impetus to be stopped. The problem is truly integrating new decision points into the traditional process and accepting new paradigms for gold standards in tox. So thank you, Bill, for that comment. And certainly anyone else who has responses or questions or comments, don't hesitate to send those through. And meanwhile, I'll just move on to question number 3, where we have just over 10 minutes left, so hopefully we can get through all of the questions as well as comments and questions coming through. So moving on, again, I'll address this.
I actually think the question from John Senior at the FDA, or the comment from John Senior, a comment and a question, is actually very interesting. And he points out that for liver industry, both at the agency and in the industry and even in clinical practice, we have many good biomarkers for liver damage, ALT, AST, etc., and they've been used for quite a time, but there really hasn't been a strong or robust scientific validation as we now insist or we expect for all new biomarkers. And his fundamental question is, well, who should perform that validation of these traditional biomarkers? So we actually have some reference data that we can point to as to how good are these ones that everybody believes is useful. And the reason I particularly like that question, if I can divert just a second, was early on we were doing some database analysis looking at tox data systems, and one of the questions we like to ask was how many of the GLP tox studies that you ran over the past 5 years did you see increases in liver enzymes that were confirmed, or where liver tox was confirmed in the histopathology? So it's a great question to actually test the power of your database and your database search engine. And of course, what people were surprised at was when companies like, shall we say, Roche, or just as an example, or another one was GSK, when they asked those kinds of questions, was surprised that there were a lot of instances where increases of liver enzymes came up. and did not get confirming histopathology, and the percentage in some cases were like 50%, and therefore across all of their studies, if one were to just do a pure mathematical assessment, you'd have to say that increases in liver enzymes were only 50% predictive and I'm not making a scientific case here. I'm just saying that people were surprised maybe that they were seeing liver enzymes increases much more often than they were seeing the histopathology. And so John's point's a very good one, that we do have these traditional biomarkers out there that maybe the degree of validation and the degree of scientific interpretation isn't what we all think it is. And I think it's a really neat question. Okay, so we can move on.
Great. Thank you, Ernie. Okay, we'll just zip to the next question. And I think you covered the first part of this, which is what safety biomarkers are commonly used and trusted today. But I don't think we've really focused much on what emerging technologies are showing promise. And I wonder if you could comment on that, and maybe in a related question that came in from the audience, is the use of microRNAs or gene expression assays, specifically for drug-induced tissue injury. So if you could talk about emerging technologies that you're seeing and microRNA.
Well, that's a question. Unfortunately, I could spend way too much time on this one. There's no doubt about it. Some of these metabolomic technologies, and they are in vivo studies, or most of the data is available from in vivo studies, although there are some new companies out there looking at in vitro, particularly stem cell metabolomic results that I think are very interesting. So these looking at the metabolome from certain organs, particularly the liver, which tends to be a very big chemical powerhouse, and therefore looking at the metabolome for liver issues tends to be, I think, right now, shall we say, undervalued, that people maybe aren't leveraging as much, but it's another one of the things that right now is kind of expensive, and therefore there needs to be some kind of balance made in there. But I really think that the metabolome, would you call metabolomics an emerging technology? Probably not. It's been around for quite a while. But in terms of your ability to use it on a routine basis, I think that's probably an emerging technology, and one that I believe has real power, and for certain toxicities, particularly, as I said, the liver and maybe cardiovascular effects. And the translation of that now into the in vitro world, I think, offers real promise. And as I said, there are at least a couple companies we know of that are promoting products in that in vitro metabolomics using stem cell or iPS cells that I think will be value added in the near term. So I'd give that as my number one example of something where I think is really coming and will be value added and adoptable.
Mm-hmm. Great. Okay, well, we are running just only about 5 more minutes. I do want to go to sort of our final question, and it also is, I think Ted from the audience sent us a question a moment ago, and he must have been reading ahead to discussion point 4. His question was, what do you consider the most significant challenge or bottleneck in the successful translation of a biomarker to a clinically relevant biomarker? And which sort of mirrors our discussion point on the screen here, which is basically the shortest path to adoption of these technologies, and the hurdles involved. So if you could, again, maybe touch on a few of those points, Ernie, that would be much appreciated.
Well, of course, we at CHA have tried to really promote the idea that industry consortium is the most powerful way of getting adoption or getting acceptance or proving the utility of these tools. But it's not easy, and it's still expensive, and who pays and who's going to pick up the tab is still an issue because the degree of data that toxicologists need or toxicologists would like to see for the scientific validation tends to be much more robust than I think most companies realize it when they start thinking about their test in the safety arena. So this idea of what is a good validation and how many compounds or how much is enough, and what is the comparator data. It really, I don't think, can be done on a company-by-company basis, either as a vendor or as users, and really needs to be done in the collaborative space. And then I think right now, unfortunately, and too many times we haven't really included regulatory authorities in that discussion and made it part of it, and I think that needs to happen much more so than we've done maybe in the past. And so it's this whole idea of getting the consensus that I think is really problematic, and I do believe consortium is the best way to go, but how to do that is still seems to be difficult. And who pays in the end is the really big question. But the other point I would like to make is, so the scientific part of it, at least we have a vision of how this could be done by bringing multiple people to the table, sharing the cost, and coming up with a paradigm where everybody is, from regulatories through vendors to users, everybody's in agreement about how the validation needs to look. But we still have a question about the cost effectiveness, which I don't think we've really dealt with. How to prove a test is really enough value added to justify the cost, or to what the pricing needs to be in order to really adopt it widely. I think that cost effectiveness validation is really the big, shall we say, unanswered or unexplored area, that there isn't really any good examples of how to do that. Don said that what people were asking for was vendors' help in providing a cost-effectiveness justification, but I think the reason why that's problematic is there just isn't the data to really make that case. What is a cost-effective new biomarker?
Mm-hmm.
Okay?
Good. Thanks, Ernie. And I think, as all of you probably noticed, a survey popped up on your screen, as I had mentioned earlier. We'd love it if you just take just a few minutes to fill that out as we're wrapping up the webinar here today. Your feedback is really important for us to understand what we're doing well and where we need to improve in executing and promoting these events. So we appreciate it if you could take that time. And I think with that, and actually the chat screen went blank on me, so I can't see the chats that have come in most recently. And so I think maybe at this time, Ernie, did you have any final comments that you wanted to share with the group, or any?
No, well, I mean, yeah, I gotta say, the one part of the question I didn't answer for you was the even newer technologies, the RNAis and the profiling of, say, RNAis. We've seen some proposals. We know some companies are working in this area. There's actually a number of publications, or at least poster sessions on this topic. But it's something I can't address from any kind of first-hand experience, nor are there many in the industry, but it is something that looks interesting, and it looks actually more powerful and more predictive, if I can use that word, than just rote screening of the expression changes after a drug. So I actually am very enthusiastic to see more in this RNA area, but right now I'm not aware of any use or any widespread proof of concept or validation data in the area.
Mm-hmm. Okay. Great. Well, thank you for addressing that question. And it looks like on my clock we've got the top of the hour here. Again, I would like to direct everyone's attention to that survey. If you could just take a few minutes to fill that out. It looks like we weren't able to address every question that came through the chat. I do apologize greatly for that, but what we will do, as mentioned, we'll compile some of the thoughts and comments and inputs generated during today's webinar, and we'll put that onto the DSEC website, at least in a blog fashion. If not, take the discussion to the discussion board online. And so I guess with that, I wanted to thank everyone for their attention today. We really enjoyed your participation, your attendance, as well as your comments and feedback that you've sent through to us. Again, I think we could continue to go on here for another hour, if not longer. So you can expect that we'll continue the discussion, certainly within the DSEC community website, and you could probably expect further discussion in a future webinar or at future conferences. So again, thank you very much for your time. And with that, I think we'll end today's session, and we'll see you next time within the Drug Safety Executive Council. Thanks, everyone.